How has mould saved millions of lives?
In this video, we're going to learn about the production of penicillin. Before we start, you should remember that microorganisms are tiny living things including bacteria, viruses and fungi. Penicillin is a type of antibiotic which can be used to treat bacterial infections. Really important, antibiotics can only be used to treat bacterial infections. Penicillin was first discovered in 1928 by a scientist called Alexander Fleming. He noticed that bacteria he was trying to grow would not grow near a certain type of mould fungus. After lots of experimentation, he discovered that the mould produced a substance that killed the bacteria.
He called this substance penicillin, which was purified 10 years later and used to treat bacterial infections in people. The drug has gone to save millions of lives. Penicillin is still used to treat bacterial infections today, but it's not made using mouldy food.
Penicillin is produced by a particular species of fungus. The fungus can be grown commercially in a fermenter like this one. The fermenter controls the following conditions. 1. pH. This is maintained at approximately pH 6.5 to allow the fungus the optimum pH to grow. It's monitored by the pH probe. 2. Temperature. This is maintained between 23 degrees Celsius and 28 degrees Celsius, again to provide the optimum conditions for fungal growth. Temperature is monitored by the temperature probe and controlled by the cooling jacket. The fungi naturally produce heat as they respire, so the cooling jacket prevents the fermenter from getting too hot. 3. Oxygen level. Oxygen is needed for the fungi to respire. The amount of oxygen available is controlled by allowing the entrance of sterile air. Why do you think it's important the air is sterile? Pause the video to think about this. The air has to be sterile to prevent other microorganisms entering the fermenter and contaminating the fungus. Unwanted microorganisms would then be grown and the penicillin spoiled. The motor turns the stirrer to thoroughly mix the contents of the fermenter. The pressure relief valve is needed to remove excess gas and prevent explosions. For example, carbon dioxide is produced when fungi respire. Following the fermentation, the products are drained via the tap. These products are a mixture of chemicals. The products can then be purified and the drug penicillin is extracted for use. Since the discovery of penicillin nearly 100 years ago, many other antibiotics have been discovered and developed. Different antibiotics work in different ways. To learn more about antibiotics, watch this video. Penicillin breaks down bacterial cell walls. One type of antibiotic prevents bacteria making new DNA, while another halts protein synthesis.
All these new drugs and millions of lives saved are down to one person's observations about bacteria in a petri dish. Amazing.
If you liked the video, give it a thumbs up and don't forget to subscribe. Comment below if you have any questions. Why not check out our AddyESchool app as well? Until next time.